is episode 271 of the Stem Cell Podcast, Skin Regeneration, with Dr. Valentina Greco. Hello, everybody. This is Dr. Daylon James and Dr. Arun Sharma. Welcome back to the Stem Cell Podcast, where we culture knowledge in stem cell research by talking to some of the brightest minds in the field. The Stem Cell Podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you enjoy the podcast, rate us and leave a review. We're always looking for feedback on how we can improve and also for suggestions on guests. So send in your messages and requests. Today, we have Dr. Valentina Greco. She's from Yale University on the podcast to talk about her research on the mechanisms of tissue homeostasis and regeneration. We've also got our usual roundup of recent highlights and stem cell news that's coming up.
But first, it's almost time for ISR, and we'll see you in Hamburg over the next few days. Visit www.issr2024.org to see the program and plan your conference. From plenary sessions to concurrent tracks to focus session and innovation showcases, there's something for everybody. And as part of the program, we're going to be recording a live session with Drs. Kat Jameson and Davide Marotta. Join us on July 12th at 6 p.m. in Hall 4 at the entrance level to learn about all things stem cell research in space. That's stem cell research taking place on the International Space Station. We're also really excited that we've been actually nominated for a People's Choice Podcast Award in the science category. Actually, this is news to me. That's quite an honor. And we'd be honored if you would give us a vote at podcastawards.com. We're going to start things off by jumping into something not directly stem cell related, but I think has received a lot of press recently. I think we should probably talk about it. It's in the world of genome editing. And as we've discussed here on the show, we're in the midst of the genome editing revolution, starting from way back from TALEN, going to, well, I guess even before that, zinc finger nucleases, then to TALEN, which everybody thought was going to be the big thing. Remember TALEN history, ancient history, right? And then what happened, of course, CRISPR came around, and most of us associate genome editing with CRISPR, and then mostly with Cas9, although that's changing in itself with base editors, prime editors, all this kind of stuff, right? Here we have Patrick Shu over at the ARC Institute, which is this really cool research institute in NorCal, Northern California. He's also associated with Berkeley. The Shu Lab is throwing a wrench in all of this. And just as a backstory, Patrick Shu used to work in Feng Zhang's lab. Feng Zhang, of course, one of the first folks to describe that CRISPR can be utilized in mammalian cultures for genome editing. So Patrick has a really strong backstory and tie to the CRISPR revolution, and he's throwing a wrench in that revolution by describing his own, not his own, but a new modality for genome editing that is thought to answer a lot of questions and address a lot of the concerns associated with CRISPR, right? We all know CRISPR. CRISPR is this way to rapidly modify the genome using these guide RNAs, but the problem with CRISPR, and typically with CRISPR-Cas9 and other endonucleases like Cas9, is that it introduces a break in the genome, typically a double-stranded break, and then you're using the endogenous repair machinery of the cell to repair that gene, the target portion of the genome, and at the same time that's happening, you can introduce a repair template while the DNA is being repaired in the cell of interest. Okay. It's a really dirty process. It's been powerful. It's been useful. It's become clinically translated, and in some situation, Casgevy comes to mind. But it's imprecise. It's efficient in some situations, but we know it causes off-target effects, right? The analogy that I always give here on the show is taking a shotgun to the genome. So here, the Shu Lab, and the first author here is Matthew Durant and also Nicholas Perry. They're using a completely new modality for genome editing, using something that we read about in our biology textbooks all the time. I remember first reading about this in the 9th grade. And these are those, quote, jumping genes or transposable elements, right? These have been around forever. You remember reading about Barbara McClintock and the transposase and the jumping gene experiments in the corn, right, that she did all those years ago. And the Shoe Lab is basically using those transposable elements to modify DNA. It's pretty ingenious. So they're actually reporting that the IS-110 insertion sequence, which is a family of minimal and autonomous mobile genetic elements or transposable elements, can express a non-coding RNA that binds specifically to their encoded recombinase. Okay. And before I get into it, this is a Nature paper, a very high profile Nature paper titled Bridge RNAs Direct Programmable Recombination of Target and Donor DNA. And I'm not going to go super deep into this because this is very hardcore molecular biology and describing how they actually identified this transposase, this programmable transposase and recombinase. But basically, there's this bridge RNA which has two loops which encode for a nucleotide stretch that base pair with the target DNA and the donor DNA, which is this IS-110 insertion element itself. And the bottom line is, you can use this transposase system, this recombinase system, to insert cleanly, insert, remove, excise, invert large segments of DNA, okay, cleanly without introducing a double-stranded break, just like what would happen naturally with transposable elements, right? I think this is a groundbreaking study. It's modular, it's modifiable, it can be guided just like using your standard guide RNA, just like what you do with CRISPR. And it's basically expanding this diversity of the nucleic acid guided systems beyond your standard CRISPR, beyond your standard RNA interference, RNAi. Okay, so and you can insert, excise, invert, just like you do with CRISPR, but much more cleanly. But the big picture, the next step is very obvious, and we were making this analogy before the show. Remember when Jennifer Doudna and Emmanuel Charpentier first described CRISPR, they described it in the context of the bacterial system, which is exactly what they're doing here. They're describing this, programmable recombination using these bridge RNAs in a bacterial system, an E. coli system. And then in the CRISPR world, Feng Zhang and George Church describe the application of CRISPR, and especially CRISPR-Cas9, in mammalian systems. And that's when the revolution really took off, because people saw, oh man, this is not just a tool for bacteria. You can use it for iPS cells, or you can use it for editing, modifying mice or humans one day, right? That's the next step for this technology as well. It hasn't been described yet using this bridge RNA recombinase system in a mammalian system, but it is coming. I am 100% sure that the Shoe Lab and their collaborators are working on this technology and bringing it into a mammalian system. And I think that's when this revolution really takes off.
Yeah, man, the hits just keep on coming. This is the age of genetic engineering in a real practical sense. And it's the culmination of many years of promise and expectation with a few fumbles along the way. And here we are moving really ever closer, I would say, to clinical translational opportunities for this and, myriad other base editing, et cetera, approaches and modalities. Although I'll say, there's still, I guess, fundamental in terms of a breakthrough, in terms of treating everything, there's still fundamental obstacles in terms of delivery and some of the cell intrinsic limitations and like clonality and perversion of the genome, et cetera. Although it would seem will take a major offender off the block there in terms of that shotgun approach that you were talking about. But for me, more than anything, I love stories like this because as you emphasize, this is a real watershed, a major conceptual advance. And like many of the conceptual advances that came before it, it really was rooted in basic science understanding, right? Understanding the molecular biological machines there in all cells or in some cells, and then like exploiting them to be used toward whatever end, since the days of like Taq polymerase, right? We've been identifying the native capacity of these things and then using them often with like a clinical horizon, but really in the more near term, the the scientific innovation and tool sets that are built to this, that's what I can't wait to see. The opportunities that we have as scientists to look into these systems is really based largely, if not entirely, on these tools that have been generated. And this is another amazing fundamental advance, technical fundamental advance that can now be tweaked and optimized in mammalian cells, et cetera. But in the same way that all these editors and different modalities emerged from CRISPR, and even Talon, give our props to Talon. No shade. That was a big one. I'll be excited to see how this bridge system gets developed moving forward, because really exciting time to be a scientist around these amazing days.
Yeah, we talk about the tech here on the show, and this is a stem cell show. Not all the papers we talk about are stem cell papers, but think about how this technology could potentially be used in your stem cell system, making much cleaner IPS lines, right? Modifying at the zygote level much more cleanly, and not having to worry about the off-target effects that are often associated with CRISPR, just by using this recombinase system, using this bridge RNA system. And yeah, I think what you're alluding to is spot on. A lot of these technologies, whether they're in the genome editing realm or otherwise, in the modern era of biomedical research, a lot of these technologies are harnessed from nature, which is so cool. You alluded to Taq polymerase, but also GFP, right? GFP came from what is it, like a jellyfish, right? Of course, the CRISPR system and bacteria, viral defense mechanism, and here once again another transposable element-based system that's found in nature. So it just kind of makes you think about what's out there in nature in these extremophiles, or who knows what other type of animal, plant animal that it's just waiting to be harnessed to drive the next biomedical revolution, right?
Yeah, and in line for the Nobel Prize on that list, right? GFP, Taq polymerase, CRISPR, all those Nobel Prize winning feats and accomplishments. Let's see if the Sue Lab here can jump in on that. High praise. Moving into applications, we alluded to the clinical application of this, and probably the first frontier, well, certainly the first frontier for genetic engineering is the blood. Nowadays we already have clinical trials that are far advanced and I guess ready for prime time for like sickle cell, other hematologic malignancies that are based on genetic engineering, kind of like almost generations ago approaches, considering how fast the field is moving, but nevertheless, actually leading to cures. And of course, I love talking about the blood. The real key limitation of all these therapies is the lifespan of hematopoietic stem cells and the scarcity. And the holy grails are either differentiation of hematopoietic stem cells from iPS cells or self-renewal of adult hematopoietic stem cells, because they're key to sustaining lifelong hematopoiesis, right? And for the most part, they remain quiescent, only mobilized to preserve the hematopoietic stem cell pool, but also remaining quiescent so that they don't become exhausted. They want to maintain that stem cell reserve. But when they're mobilized, it's usually under stress conditions, right? And under those stress conditions, like, chemotherapy, irradiation being a cardinal example, these cells are prompted to exit the dormant state. They activate cell division. They replenish the hematopoietic pool. Now, how hematopoietic stem cells maintain their quiescence is a key goal, right? Defining the mechanisms of hematopoietic stem cell quiescence. Been countless studies, and we talked about a lot of them on the show, but it's been pretty well defined and established that mitochondria play a critical role. And emerging evidence, more recent evidence, is suggesting that this mitochondrial permeability transition pore, let's call it the MPTP, is a critical player, right? This is a gateway for reactive oxygen species translocation from the mitochondria into the cytoplasm, okay? And the increased permeability of these MPTPs has been linked in many systems to premature cell death. And conversely, if you suppress the permeability of these MPTPs in the context of hematopoietic stem cell transplant, you enhance the efficiency of that transplant, also the collection. So the number of hematopoietic stem cells, phenotypic at least, that you get is increased when you inhibit that MPTP. And also under irradiation stress, where you're mobilizing hematopoietic stem cells, you get a marked increase in MPTP activity. So it seems like the link is there between these mitochondrial permeability transition pores and maintenance of stemness or quiescence of hematopoietic stem cells. But the underlying mechanisms defining how the MPTPs mediate this effect is unknown. All right, so this was the goal of the Yu Hao's lab, who's in Chongqing Medical University in China. And their group set out to define the role of mitochondria and specifically these MPTPs in maintaining stem cell quiescence. And they use this irradiation system. And by irradiating mice and doing a differential gene expression in hematopoietic stem cells, they identified this gene ninrin, right? And then went deep, generating all kinds of mice, knockout, a knock-in, GFP, in order to define the expression profile of ninrin and its role in hematopoietic stem cell frequency, dormancy, and self-renewal. And what they showed is that under the influence of irradiation, they found abnormal MPTP opening, mitochondrial swelling, and elevated reaction oxygen species levels. And what they identify is that when they knock out ninrin, they found diminished hematopoietic stem cell frequency, dormancy, and self-renewal. And that was linked to abnormal MPTP opening, mitochondrial swelling, and elevated levels of reactive oxygen species. And this also reduced the tolerance of hematopoietic stem cells to radiation and promoted this necrosis-like phenotype that had also been observed in other systems. On the flip side, if they express ninrin in hematopoietic stem cells, they diminish that irradiation-induced lethality. And when they mechanistically, looking into this relationship of ninrin and hematopoietic stem cell quiescence and mitochondrial activity, they found that, and when they delete it, they get activation of this transcript PPIF, and that leads to overexpression of cyclophilin D, which contributes to mitochondrial dysfunction. And here we go in terms of like a therapeutic strategy. They found that PPIF haploinsufficiency or pharmacological inhibition of cyclophilin D significantly mitigated these effects. So they were able to effectively rescue the hematopoietic stem cell function in these ninrin knockout mice. So this was a deep study. I love blood studies, but it's rare nowadays that you get a study that identifies a new role for a gene in hematopoietic stem cell function and then does all the necessary mechanistic study with the knockout, knock-in to provide not just a descriptive trajectory of the role of that gene, but also its necessity in maintaining hematopoietic stem cell function. And then finally here, having the therapeutic line here with the pharmacological inhibition of cyclophilin D, I think whether or not that's really going to be translatable in a human system, given off-target effects that we always talk about, at least it identifies a system which ex vivo may be applied in the context of hematopoietic stem cell transfer or expansion to maintain stemness or quiescence of hematopoietic stem cells in like different therapeutic contexts, maybe not in the body. So a rich study from Cell Stem Cell and the Hao Lab. Arun, what do you think?
Yeah, cool stuff. I mean, like we've had a few of these studies recently on the show in the HSC realm, where we basically the authors are converging on a node for a gene or something that's regulating HSC biology, and they kind of identified that with ninrin here. I think it's a really exciting thing, when you identify a node like that, but certainly you have to think about the limitations. You alluded to that, seeing if it's feasible to actually bring these discoveries into a translatable human context. And they actually straight up say that. They're saying while these models are great at understanding, are improving the understanding of HSC biology, you have to be careful when extrapolating these results to human biology, because of course humans are not mice. So they got to do the validation in the human samples, looking at species differences. That's going to be really critical for making that translational jump, right? I mean, kind of like what we were talking about in the previous story with the bridge RNAs. You can demonstrate these things in the model system, but going into human is a whole different beast, right? One other thing to think about is the duality of the role in NINRIN, and not just protecting healthy HSCs, but also potentially protecting leukemic stem cells too. So
Mm.
how do you ensure that the cells you're protecting are just the cells you're protecting are just the healthy ones, and not the tumor the tumorigenic ones, or the cancerous ones, right? So I think there's a lot of doors that have to be opened before this can really be brought to the clinic. But really cool to see a particular node in this realm of HSC bio. Any paper that we cover that comes to a single focal point, I think is really exciting, because it becomes an easy way to modify these HSCs just by targeting that single node, right?
Yeah, I mean, we talk about single node and holy grail and all these ideas and symbols that are kind of convey a silver bullet type approach, like there is one thing. And if anything, I've learned just doing the show. I mean, we've talked about many of these stories which identify the role of a single gene product in mediating a critical function of hematopoietic stem cell or other cells even. And the reality is that there is no silver bullet and there is no single player here, that it is a multiplicity of factors, down to the organelles and their activity, that are able to balance the intrinsic capacity of the cell with the extrinsic or environmental cues that it receives on the outside. And of course, these systems have been tuned over eons in order to work within these boundaries. And in order to breach them, we're going to have to really, understand them a lot more closely. And this is a step in that direction. But like many of the pharmacological compounds or single genes that have been implicated and held up as the answer to indefinite hematopoietic stem cell self-renewal or clinical potential, they've all fallen a bit short. But we are moving closer. We are moving the needle.
Yeah, that's for sure. I mean, don't want to say this is a magic bullet or silver bullet, whatever you want to call it. It's one of many, perhaps, targetable nodes for regulating HC function. And like I've alluded to before, maybe we have to take a combinatorial approach to all of this and really generate some sort of cocktail or whatnot. Moving on to something related, this is an engineering application for macrophages. Primitive macrophages enable long-term vascularization of human heart-on-chip platforms. This is coming from the Melissa Radicic lab over in Toronto, who has been working on these heart-on-chip approaches for a long time now. I mean, she's a bioengineer, has been working in the area of cardiac bioengineering for about a decade plus. Other folks on this particular paper, which is a cell stem cell paper, include Gordon Keller, the one and only Gordon Keller, who's been involved in so many different applications of differentiation from pluripotent stem cells over the years. First author here is Shira Landau, as well as Yi Zhao. I've been working on these hard on chip systems for a little while myself, nowhere near as long as the Radicic lab has been working on it. But there's these really cool bioengineered systems that you can utilize to make more mature vasculature and also make more matured cardiac tissue just by integrating this tissue, stem cell derived cardiovascular cells, into this bioengineered chip system. This system is sort of similar to the one that I use in my lab, where you can add the cells and then you can biomechanically stimulate the cells on the chip by introducing fluid flow. And we know that endothelial cells really like to be perfused and respond really nicely to shear stress, further inducing their their maturation. And so the rationale behind this study, I think, is relatively straightforward. We want to make the myocardium, the cardiovascular tissue of the heart, the muscle tissue of the heart, we want to make it more mature. And part of that is making the vessels more mature too. So the myocardium is complicated. It's got multiple different cell types, biomechanical stretch and stimulation going on during every single heartbeat. And you want to see if you can sort of replicate that using this chip-based system. And here, what they saw, building on some previous studies with in vivo neonatal studies, they actually showed that there's a role for resident cardiac macrophages in inducing a post-injury regeneration and angiogenesis. So there's these macrophages that are in the vasculature of the heart, and they have this somewhat protective effect in an injury-based system. And it perhaps is a bit of a stretch because we know that the human heart has a limited regenerative capacity. But that's something that they wanted to build on here. So here, what they were integrating pluripotent stem cell derived yolk sac-like macrophages with a vascularized heart-on-chip platform. Okay, and what I like about the study is that you can really look at the modularity of it, right? If you just have the endothelial cells that are being grown on the chip, they don't do a great job of turning into those vessel-like structures. But if you keep on building different cell types into the system, you have the endothelial cells, you have the macrophages, cardiomyocytes. With every single cell type that you build onto the system, the vessels become more stabilized. They become more mature, which is ultimately what you want for making these hard on chip systems, anything on chip systems, better and more stable long term. I mean, this is something that we ran into in our lab as well. There's an upper limit in terms of how long you can actually grow these cells on these organ on chip systems without the chip just kind of falling apart. These cells aren't designed to be long term on these chips. So just by introducing these macrophages, they were able to reduce the overall tissue cytotoxicity and release of cell-free mitochondrial DNA, while actually upregulating the secretion of these pro-angiogenic matrix remodeling and cardioprotective cytokines. So that's great. And then they did a bunch of RNA sequencing, single nuclei sequencing, to show an upregulation of what you want, which is that cardiac maturation, upregulation of angiogenic genes, also suggesting that these macrophages might prime stromal cells for further vascular development by inducing an IGF binding protein 7 and hepatocyte growth factor expression as well. The hepatocyte growth factors expression to me is still a little bit confusing. Like, what is that doing, in this context? But ultimately, it's a cool system showing the importance of different cell types, in particular these primitive macrophage-like cells, and actually the long-term vascularization of cardiac tissues and these cardiac tissues on the chip. And the application of these chip systems is sort of obvious. If you have more mature cardiomyocytes, more mature cardiovascular cells, you can use them for better disease modeling, better drug screening systems. But like what we've actually talked about with some guests here on the show who have worked in this area, scalability is still a bit of a concern, because how do you kind of balance the more mature system with something that's more mature but also can be scaled to like a 1536 plate format that you can use for drug screening assays? So a lot to think about here, but I think a nice cell-based approach for driving maturation in these chip-based systems.
Yeah, I agree with you there about the my fundamental takeaway. The exciting thing I see there is, it's a cell-based approach, the biology of the cell presumably driving the biology of the rest of those organ components on the chip. I like that. And it's all ES derived, right? All IPS derived. I like that component that you don't have to pull cells from elsewhere. But I think there's a challenge for me just conceptually. It's a little bit of a conflict, right? As you were saying, getting more mature by using these primitive yolk sac, right? Macrophages. So it's a question of like, well, are we recapitulating like the physiological cardiogenesis or the components that are present in a physiological heart? And yeah, maybe not. And does that even matter? No. I mean, the tool is as good as its application and what insight you glean from it. So I can totally I get that and I accept that, but still I have some questions as to how you would use it in that case. Because one of I think the most exciting things that you could use these models for is to try and interrogate like cellular events. Like you were talking about the rationale for this being that macrophages in part mediate the post-injury angiogenesis, but like they also mediate the scar formation, right? So I would ask that question right away. It's like, okay, is this close to a physiological model of ischemia reperfusion injury in that the macrophages can have this pro-angiogenic quality that we observe, but do they also have this, I mean, potentially maladaptive quality? So effectively, like while this is a model, the hard on chip is best used as a model. I think it's important to explore what the limitations and or applications boundaries of that model are. But fundamentally, I love the fact that they're adding another cell here and showing that it really, in the A/B testing, has a significant influence on vascular morphogenesis and the maturity, and raises questions as to whether or not a similar approach could be used in other kind of tissue organoids.
Yeah, I totally agree. It's perhaps a cell that you wouldn't have thought of that would be influencing this role, but maybe it is because, like you're alluding to, macrophages have a strong angiogenic effect in terms of vessel remodeling and all this kind of stuff. It's a I think there's a lot, and I think you're getting at this. There's a lot of mechanism that is missing in this study, like and I think they actually are very upfront about that. They think there's a lot of mechanistic insights that have to be gleaned from actually like figuring out how these macrophages are having this impact, right? It's more than just adding cells and seeing the downstream. You want to understand why, and no doubt that they're starting to dive deeper into that and teasing apart those cellular mechanisms, right?
Absolutely, and I think at the end of the day, the real key here is that the sophistication of these models for understanding, the interrelationship of cells is only increasing, and it's something that nowadays is actually being applied. I think the new medicine is really appreciating patients on an individual level, and that's our final story. The Roundup actually leverages brain neural organoids toward that purpose. And it's based on this paper. It's a Nature paper from Parla Arlata Lab. Is based on this idea of genetic variation and susceptibility to disease triggers, whether they be environmental or intrinsic. Um, and the challenges to that, which is that the models that we have had in the past that were scalable, right? Essentially like, monolayer culture, they've been very effective. Yeah, there's been stories from Loren's lab, Egan group that have looked at 2D neuronal cultures to produce these kind of population scale models, a kind of village in a dish using many, many donor cell lines. But of course, as we've talked about now for too many years, the 2D, who needs it? Anything 2D, I don't get out of bed for that. But in the age of organoids, it's a bit of a challenge to extend the scale there um, to a 3D approach, right? They can more closely model the cellular complexity and the developmental events in the brain. Um, but when you look at patient to patient, it's hard to get a defined standardized approach. You look a lot of these protocols and it's like 2 to 3 days, 8 to 6 days, 8 to 12 days, because there's a range there. There's differences in the growth rate of the cells, just like proliferation rate basic, also the pace of differentiation. And particularly when you have multiple cell types, like in these organoids, that are co-developing over extended periods, as you were alluding to there, Arun, in your story about the heart organoids and heart on chip. It's hard to get these things to go for a long while. So in this story, which I think is a tremendous innovation, uh, it's straightforward, the approach here. Our lab introduces what they call these 3D brain chimeroids. Okay, so these are multi-donor chimeras or organoids made from multiple donors, as many as 6 donors. I think we're using this paper in different combinations with like a relatively balanced representation of cell types across those donors. And what they found is that when they put them all together, they start from like either neural progenitor or neural stem cell phase, and they combine um, cells from multiple single donor organoids and find that ultimately, when they let those things go, each of those donors accounts for all cell lineages of the cerebral cortex. And then they use this and apply this, and this is the fundamental rationale for this, is that that adding them all together will kind of revert them all to this mean level of developmental timing so that you can look at all these cell types at the same time. They can monitor them on the back end single cell sequencing by looking at, reference to the whole genome sequencing of these same lines to see which cells came from where. And they showed that there was a variation, a fundamental variation in the susceptibility to ethanol and this anti-epileptic drug valproic acid. And these are two neurotoxic triggers that are, have been shown to be highly variable amongst different groups. So they showed that, yes, they verified that using these 6 different donors, that there was an individual susceptibility, the variability in the penetrance, and also the actual phenotype at the different cells. Even though they were in the same organoid, they displayed different phenotypic response to these insults. So I think this is, we talked about the bridge thing to start transposase, like watershed. I don't want to put this in the same category as that, because I really think that is going to, change the game. But this also, I think, a fundamental technical advance that is going to allow a lot of differential analysis within a combined system that is then scalable. And you can look at multiple. I mean, I think you could scale this up beyond 6, even with the way the genomic sequencing and tagging and all those methods are improving, where you can really look at like village in a dish, cut that down to like village in an organoid, or at least in groups of organoids, and across many different tissue types beyond just the neural organoid. So a really exciting technical advance here that I think is going to bear a lot of fruit.
Yeah, 6. I don't know if you would call 6 a village. I think 6 is like a family that lives in a village. But yeah, if you want to turn this into a village, you gotta up the number a little bit, and inevitably that's what they're going to be doing here, going for hundreds and hundreds of perhaps different genetic backgrounds integrated into a single organoid. But then you start to wonder about organoid size, and you got to figure out ways to keep these things intact. There's some inherent issues with large organoids, at least when it comes to their necrosis. And so I think we have to answer some of those questions too. But I think a really cool system, these cell villages systems are, I think, really exciting, and in part are enabled by the tech, enabled by the single cell sequencing, the single cell analysis that you can utilize to evaluate how the individual genetic backgrounds are responding to, say, developmental stimuli, or in this situation, neurotoxic stimuli like a drug response. My lab is actually also kind of working with some groups over at UCLA, like Michael Wells' lab, to explore this in a 2-dimensional context. We're not turning these things into cardiac organoids, but potentially looking at patient-specific drug response using a bunch of different cardiomyocytes from different genetic backgrounds all pooled into the single population. So these are really cool technologies that are enabled by the intersection of multiple technologies. These chimeroids are enabled by advances in single cell RNA sequencing. You got to get around some of the limitations with the organoids themselves, but I'm excited to turn this into a village, or excited to see this become a true village, right?
Yes, I mean, 6 is a small village, but it's not unprecedented, Arun. I mean, you city boy over there. But I think the what you're saying there is true in terms of limitations. How many different donors can you get into a single organoid? I get that, but I think that we should also, based on that, we should also highlight. I think one of the maybe the more important biological advance here which is that when you combine, I mean, this is the thing, you're comparing one organoid to another, a single donor in the past, and you have to wrestle with that idea of like the pace of development. And are you even comparing, you know, apples to apples? And here, I think the key finding, which, you know, they had to do it to see, and I think it's an amazing proof or support of how the system can be applied, is that they all kind of coalesce onto a single, I mean, there's going to be some variation, of course, but a relatively single developmental trajectory. And I think that that is the key finding here, is that, yeah, sure, you can only put so many cells in an organoid, but if you can put 20 even, the fact that they will all align to that same pace means that you can do 20 in one well, 20 in another, 20 in another. And, you know, as you talked about, the technological advances will facilitate an approach that will actually approach the scale of village that you're accustomed to over there in at Cedars. Well, it's an interesting day for technical advances on the show, but also really propping up the future insights that are going to drive innovation in the clinical space. And we're going to see some of that at the ISSCR when we get there. Can't wait. Before we move on to our interview with the next president of the ISSCR, I have a quick message from STEMCELL Technologies. Interested in exploring the applications of organoid cultures? Get a free wall chart showing how organoids are used as model systems to study infectious diseases, cancer, congenital disorders, and tissue regeneration. To learn more, visit stemcell.com/organoids-modeling-wall chart. All right, everybody. Today we have a very special guest, a great friend of the show and future president of the ISSCR. Also, the Carolyn Walch Slayman, professor of genetics and co-chair of the Status of Women in Medicine, or SWIM, at Yale University, Dr. Valentina Greco. The Greco Lab's goal is to define how tissues maintain themselves throughout the course of our lives in the face of continuous cellular turnover, frequent injuries, and spontaneous mutations. Ongoing projects are looking at understanding the mechanism of skin tolerance to mutations, as well as how the different tissue types, such as epithelium, fibroblasts, and vasculature, contribute to skin homeostasis throughout our lifetimes. Valentina, thank you so much for joining us today.
Thank you for having me.
Yes, welcome back to the show, Valentina. And if you can believe it, it's been almost 4 years since you were last on, which was episode 172. And today we're actually 99 episodes later at episode 271. Great timing. It's an understatement to say quite a bit has changed, since the past 4 years. For one, last time we had you on, we were in the middle of something called the COVID-19 pandemic. Almost seems like a distant memory to some of us. In terms of the lab, in terms of your lab, you've had a number of awesome publications on topics ranging from KRAS and ERK signaling to hair follicle biology. And of course, we'll talk about your role as the soon-to-be ISSCR president in a little bit. But for those of our listeners who weren't around 4 years ago, could you give us a broad update in terms of what your lab is currently working on?
Absolutely. And I'm glad you connected to the pandemic. First of all, it was a ray of hope connecting with the two of you during the pandemic. It was those moments where we all felt isolated, and being connected at that time with strangers and thinking together, it almost brought me a sense of belonging. And from that moment, I feel like I feel a particular peacefulness looking back, because when looking at my lab in that moment, all I felt is that I wanted people to look back today at that time and feel like somehow they managed to be on the other side of it. So the publication you mentioned to me, for me, speaks about the human infrastructure behind it, the emotional taxing, the physical efforts, but also the lost feelings that we all had to navigate to figure out, is this still our purpose in life? Do we still want to do that? What does it mean to us, and what cost is coming? The large majority of my lab is immigrants, so am I. And so for all of us, it was also this sense of longing to the place we come from, wanting to be anchored back, not being able to see our family. So seeing all this publication that we fought so hard, like everyone, and I'm sure with a lot of more privilege that I like to recognize, having these 8 papers going through the pandemic and being able to now materialize between last year and this year has been, short of magic to experience.
Yeah, I mean, I love the model, the skin and hair follicle. They've emerged as arguably the most versatile and expansive models of stem cell biology, encompassing both the cell intrinsic mechanisms as well as these niche-driven behaviors. And they are attractable, right? And visually stunning, the data that comes out of these experiments. So much so, I would argue that, the model has become the kind of avatar of stem cell biology, so much that maybe the clinical translational impact of research in skin and hair is not highlighted enough. So I'd like to take a step back and then into the future a bit here. What are some of the biggest ways, in your view, that the science of skin and hair follicles in the last couple decades have moved the needle on disease and cancer and, baldness even, asking for a friend there?
Thank you. So first of all, thank you for all the compliments to the field. And I would say that any field could provide exactly that when people come together with complementary approaches to solve questions. I also build on the shoulder of an entire field that has constructed our understanding in the skin, from the epithelium to the follicle, but also all these dermal components, as well as the field of developmental biology. They continue to innovate with microscopy and approaches that allow that visual approach also to come to place. And in terms of disease, I think we started many decades ago with the idea that we can put these cells in culture and repair burns, right? Howard Green, for instance, work that has certainly charted the way that in that manner. Also, we can shift to cornea and see all the innovation, including also gene therapy in the skin. The Graziella Pellegrini, Michele De Luca have been really in the past for so long. And there are many, many colleagues that have not only allowed basic research to flourish, but also to get translated in a way that we actually see already in the hospital.
Yeah, you alluded to the importance of imaging and how your lab is really using imaging to its full potential. And we actually had your fellow Fuchs Lab alumnus, Sherry Cohen, on the show not too long ago from UC San Diego, just down the road from me. I mean, she's also doing this exquisite work on understanding the lymphatic system's role in regeneration using, again, some gorgeous imaging approaches. And your work certainly shares that same affinity for these incredible high resolution imaging modalities. I mean, it's a common thread that I can't help but notice among all Fuchs Lab progeny is this emphasis on the visual. I mean, is this something that Elaine really emphasized back in the day, or is this just a critical requirement for the work that you do? So just, talk to us about that, the power of the great visual. I mean, how your lab is able to use this high resolution imaging to its full potential.
Yeah, thank you for the question. Maybe stepping back, I feel that we human have always moved forward in rationalizing what we go through storytelling and visual. And so I think it's something that may be relatable for many of us. To my own chemistry is such that movement, colors, and shape is really what motivates me. So imaging certainly has an impact, but it's also a source of joy for me and discovery. So much that, for instance, the lab has experimented now to create processes by talking to colleagues in the art and going to museum and thinking how curators can really make us extract information from painting in a way that creates much more informational depth. They were using this methodology for ND, PhD students training because they were noticing that in the clinic, they may be stopping too soon in diagnosing patients. So imagine how visual now becomes so really connected, not only in our capacity to relate to each other, to get excited, but to get cured, right? Because then you can get past initial assumption that you might be making. So I think the visual is both an intrinsic chemistry, but also a rewarding one externally and a way to connect to others, but also such an infinite information in all this pixel, in all this signal that you collect, that if you bring diverse lenses in it and you leverage your own experiences of any life work that you had, you can really start to see things that then in a reproducible manner you can go on to demonstrate. So this is what imaging is to me. Where do I take it from? Many of us take it from developmental biology, has been for decades innovating from any model organism you can dream of. And also in the field, for instance, people like Shosai Yoshida had already pioneered imaging to photon and confocal for testes, Christina Lo Salso together with Charles Lin and David Scudden in the metaplated stem cells. So I do believe that is an application you can do to many tissue, blood, things that you will intuitively not think that can be imaged. Of course, they have niches. They have a home where they reside that could be tackled. And in fact, people like Paul Frenet, Sean Morris, and many others have started to look at that also through imaging. But I think that the tissues that have a solid structure may be intuitively through pathology being inserted in the field as more tackable with microscopy, because you can put an H&E, put it on the microscope and start to see things that don't make sense and therefore allow for diagnosis to be accelerated. So in summary, I think some tissue may be more prone to certain technique, but I think the combination of all these techniques across all tissue would allow us to combine something that is so intuitive for many visual with many other deeper mechanistic analysis that can be combined together.
Yes, I mean, I always lean on the visual, but Arun and I talk about all the time that observation takes many forms, visual maybe being primary, but there's all the observations, the indirect observations, they're also so critical to science, and the combination of them is how you reach a conclusion, right? But I mean, at the root of it, right, from day one, Anton von Leeuwenhoek looking down the lenses there, I'm sure he was like, holy whatever, and whatever they say in German. This is a family show, but you know, I think that's the awe of seeing something for the first time or not for your first time, and just being like, wow. And you know, as I'm quoting now from a paper you just recently published that you just alluded to, observation is the heart of research, and I totally agree with that. It's a ground truth of particularly developmental biology. And like I said, that was the opening of the abstract for the paper you just published with Linda Friedlander, who's the head of education at the Yale Center for British Art. And this was published just recently in the journal Development, which I just love. I love that there's a story where an author from a curator effectively is on the author list for a development paper, because it's been a long time coming, you know, not your typical collaboration, but somehow totally consistent with your unconventional thinking and outside the box approach to problem solving. It's why I love your lab and your mind. You really do things in your own way, in a special way that I think a lot of people could take a lesson. And just to drill down a little bit, in that effort, that paper you're talking about, the lab members first practice looking at paintings while separating the act of observation from interpretation. I think that's such a simple, straightforward idea, but I think it's so key to how we go wrong in science, is where we've already interpreted it oftentimes before we even see the results, because we find what we're looking for. And here you showed, like in a scientific way, that applying that approach to microscopic image, you can draw out additional data that may have been overlooked. But, you know, I don't want to get cynical or, you know, doomsday-ish here, but there's a major sea change in the art world, also just the world generally, in terms of the rapidly advancing ability of AI to create, right? And while we've been in science, we've been using machine learning, AI, for analysis for a bit here, I think a bit ahead of the ChatGPT curve there. But it never occurred to me to think of that machine as observing the experimental results. I always assume I was doing some analysis, computation, but it really is observation, right? That's exactly what it's doing. So it's just, again, not in a doomsday way, but what do you think the limits of AI in science are, if there are any? Are we like replaceable as scientists? Is there going to be a takeover by Skynet? What's going on?
Yeah, it's a cool view into the future. I think as long as we want to talk to each other, of course, replacement cannot happen. And if AI will talk to AI, then maybe. But I think what the AI could do for us, but I'll tell you what I wish it will do for us, is to optimize the time investment into extracting things. But what I notice is really our social construct that will determine what we'll latch on and what we want. To take, for instance, the way that we have looked at oncogenic mutation, we have learned so much that when you change that genome, Varmush work and many others, that you do create some cancerous opportunity. But then Abyssal kept teaching us that you take those same cancerous cells, you put it in an organism and nothing happened. Right. So it is always that reminder, like you were saying, skin does give us an opportunity, intrinsic and extrinsic. Well, I would say any of us is an expression of that, right? Like all my membership are an expression of what I've been allowed to do and what I've been able to grab, even like if it was my right, and also what I've not been allowed to do. And so that combination of intrinsic and extrinsic and the capacity for us to figure out and be tuned, AI is spitting all these. Which one am I going to be tuned to is still in our own capacity. So my desire for AI is to actually figure out in what way we have color the way that we do our profession, in seen only with the very minded of very few in power, of membership that we know are more accessible to resources. My own whiteness makes me so close to power resources, but also my femaleness makes me underprivileged. And so how do we understand that struggling privilege as this power construct for which AI could allow us to now fish out results that we wouldn't see, because we tend to see the world as a scarcity workplace, as a winner or loser. Well, if it was much more horizontal, there were many opportunities of integration of multiple truths all at the same time. AI could allow us to actually be much more integrated, inclusive by the real meaning.
Yeah, it's really an interesting philosophical question. I mean, the human element in AI, because it's more than just a tool. It's perhaps it's a new way that we can see the world, and perhaps a new way that we can see ourselves as well. So we'll have to think about that a little bit more. But shifting away from the philosophical for a little bit, I mean, something a little bit more tangible, I suppose. We've been highlighting the upcoming ISR 2024 annual conference for a few months now here on the show, and it's really hard to believe that we're all going to be in beautiful Hamburg, Germany, for the conference in just a few weeks now. It's, of course, a very special time for you, as we alluded to, as it marks the beginning of your term as the new president of the International Society for Stem Cell Research. So congratulations. Becoming the president of the world's leading society for stem cell research is one of the biggest honors that a stem cell biologist can obtain. And we're very excited to see where you're going to take the ISSCR under your leadership. So could you give us an overview of maybe some of the goals for the next year as the new ISSCR president, what you're hoping to accomplish?
Yeah, so anything we've done in history of life is a group approach. We love to think about our own individualism as impactful, but first of all, unless we are synchronized with others thinking alike, things won't happen. And second, really, there is no way to reach a goal for others if it's not with others. And so, first of all, I follow Amanda Clark, incredible leadership that was inspirational to follow. I found Lynn before that. And together, we have been working for several years. The presidency is 1 year, but you start 2 years ahead with vice president observing and integrating what was, for instance, coming out of the strategic planning. It was based on the efforts of many board members that have worked hard at that. And so integrating those shifts in figuring out how do we approach more of a grassroots approach. So, for instance, this was I found presidency trying to understand what have we missed that the membership is really asking us to do, because decisions are often in a closed door of very few people. They tend to be kind of homogeneous in thinking. Second, thinking about involvement of how do we think about accessibility. This is Amanda's commitment to understand our efforts accessible to everyone. How do we include more parts of our world? And how do we continue to really be true to our name is my question. We are called international, and yet we know we struggle of being very strongly represented as US, and often the membership gets also redefined as white and male. And so these pools are not to be looked as like if we need to feel bad or being good or bad people, but a strong commitment that we each of us need to make in transforming ourselves to want everyone in the room. And so how do we go about one, acknowledging all the human infrastructure from ISSCR, incredible staff that continue day in, day out to innovate and listen to that membership of grassroots movement, to all the scientists also that are outside ISSCR They keep coming in and do this voluntary work on committee. How do we acknowledge it? How do we bring it out? How do we nourish it? And then how do we make sure that we vet processes by which people are coming in? Are we having processes that really are open to everyone, that are inclusive, that as people come on board, they are supported for the time they put in? And how do we keep understanding the insertion of new voices and new generation? In any organization, we struggle because, first of all, there are people accustomed to be present, and so they can tend to be tapped more. But the more we are in the organization, also, we are still aging. And so it becomes really an established, maybe chronic way of thinking about problems. And we just said before that if we don't have multiple lenses, including radical young one that says, we might be getting it all wrong, we are not going to innovate and really serve everyone in the future. And so having processes, I think, that can be not only scrutinized but improved to withstand the passing of time beyond my presidency, that is really endorsed and practiced by everyone, that's really something I'm passionate about.
Wow, your passion comes through, and you've clearly thought so deeply about it. And I really admire the leadership of the ISSCR over the last 5 years, longer. I think for a long while diversity statements have been across the board, kind of like a prerequisite PR across many industries, and they're there because they have to be there. And you don't really know if the culture of the place really has integrated it or is committed to it. It's so clear at the ISSCR that the leadership is committed to the idea. And that commitment, I think, stems from the recognition, as you started that answer, saying that, yeah, that the population at the ISSCR is skewed, and the population is skewed just fundamentally by a lot of extrinsic forces of who has access, et cetera. And I think that's the first part of real change, is recognition. So I admire the ISSCR for not only recognizing it, but committing over the years. Because, yeah, the world of stem cell biologists is big, and it's expanding. And with that expansion, the pace and the advance of stem cell research has been ever swifter. It seems to be accelerating. And nothing illustrates that more than the human embryo models, right? Introduced to the public at last year's meeting and applied in scores of labs across the globe at this point. Human embryo models have revolutionized our understanding of peri-implantation biology, but also really raised questions, I would argue, re-raised questions about the ethical implications and limits that should be enforced. And ISSCR has been way ahead of this, from long experience. I think they anticipated this and released a statement supporting the work under strict guidelines that ensure the rationale is justified, rigor, oversight are assured, limits are imposed, more. I really refer everyone to look at those regulations, because they're clear, and I think they provide a pathway versus the NIH, which is not going to support, which is okay. They're not going to support and provide funds for creation of human embryos for research, but it's the way in which they do it that I kind of am disappointed at. Embryos here are defined as, quote, derived by fertilization, parthenogenesis, cloning, or any other means from one or more human gametes or human diploid cells, unquote. So that's pretty much saying under any circumstances. I mean, there's nothing that doesn't fall into those criteria. So that's a very inclusive and blanket ban on funding. It doesn't seem like it's going anywhere. Of course, the work's going to go on. But with abortion rights vanishing, and now you see evangelical Christians in the US are circling around fetal personhood, I fear a return to like the violent, maybe that's a strong word, but it seemed to me at the time, violent rhetoric of 20 years ago, roughly, before iPSCs provide an alternative to blastocysts or SCNT for generating pluripotent stem cells. So you, as a highly visible advocate for the work, how do you strike a balance between enabling the science as you do, but while also navigating this fraught public relations element and governmental, oversight element and regulation? How do you find the path?
Yes, very important, very complex, and hits deeply, right? Because you're speaking about a movement also of the limitation of freedom of women, that then will lead also limitation for sexual orientation that are not heteronormative. We're seeing the path is, and even with the reference to DEI, you know, they were going into yet another dismantling that is active. So how do you, from the social framework, go into a research championing, nourishing, and protecting area of research that are important? First of all, as we have seen this arising, Amanda has done an amazing job together with the policy and the guideline committee, not only on creating the guideline you refer to, but also going and in the White House and trying to enter in conversation with people in power that they can be educated further, people that are already in line with us, but they need more education in order for them to champion, to counteract, and to create the resistance that is needed. I think overall, what you're bringing up is at any time in any field, there is a need for us to think ethically, because this is coming just up against what we are discussing, but there are many other areas where things can turn very quickly. So how do we go about championing the research that we need to do, creating a space, helping NIH to make right decision, another entity goes into some of us championing the conversation as part of our role, the presence as a part of our role. And it is difficult in an industry where, you know, we are rewarded for the money we bring in and the papers we bring in. So the people that often they want to do this job are also not rewarded by the system and might even lose their jobs. So it's a complex matrix. And I think ISR at the very least, but many other societies have done that too. American Society of Cell Biology forever has been championing issue that way, is together across society and together within society, how do we go and create the resistance that we need?
Yeah, I think it's like we were talking about, the ISR has really taken the forefront in trying to tackle some of these ethical issues and address some of these concerns head on. I mean, you see the guidelines coming out and being updated every few years. I think for a decade plus now, they've been trying to really, enable this conversation, not just among the scientists, but try to make these guidelines accessible to the general population, which ultimately could also be affected by some of these technological advances in our field. So, I mean, it's a major part of what ISR does. And I mean, indeed, the society has had a really special influence and importance in your career, your scientific career over the past 2 decades. I mean, you've been involved with ISR for a while now in various capacities, have been honored with pretty much every honor that the ISR can give a stem cell biologist, ranging from the Outstanding Young Investigator Award in 2011 to the Momentum Award 10 years later in 2021, before, of course, becoming vice president and now soon to be president. I mean, you've really done it all within the world's leading stem cell society. So could you just reflect on what the ISR has meant to you, both personally and professionally, and maybe take us back to when you first got involved with the society as well?
Yeah, so in 2009, I remember, I don't know if it was my first ISR, but I remember to have received both a travel award by New York Stem Cell Foundation and also to have a talk. I remember to stand on that stage, trembling, giving the talk, not receiving many questions, but hey, I was there. And I remember that as I kept coming, even though I didn't have any talks, I felt that people were willing to have a relationship with me. And by people, I mean anybody, whether it was vendor, trainees, junior faculty, or even senior established people. The key for me is also to reflect on what it means from the names you mentioned, the labs and PhD and postdoc. Those are also a label of approval, right? So I understand that my experience is also color from my legacy, the way we like to think about science, that has supported my disability, even though I might not have had thoughts. So I wrestle, right? Like, I've had an incredible experience, but are we all having the same experience? How do we provide to all of us the same experience? What does it mean to have certain labels versus not? And my observation is that even though at the beginning, of course, like everyone, I was throwing so much out, as soon as one thing started to come in, it was like a circle of reinforcement. So my observation in the profession is, as people get on the other side of something, then all of a sudden it becomes a self-fulfilling prophecy, whatever it is. And so how do we also, certainly celebrate when we get those things because the ego gets struck, but how are we gonna go to be more reflective on who gets put up for those awards? Who is that committee composed of? Are we actually creating committee they can see everyone. And for seeing everyone, it means each person that committee needs to span a broad range of membership for us to be able and empowered to nourish and cherish each different membership. Because those power forces that we speak about are in the room, whether people are represented or not from that group, because we are all oriented to power. And so even when I was celebrating people here in this podcast live, I started to think, wait, did I already skew towards that group? And I'm thinking, why didn't I say that name? Why didn't I say that name? So I get myself caught all the time. Am I fitting an expectation? Am I speaking from my heart? How do I do both so that we can be truthful and empowering of everyone and not just as minor group? So yes, I've been celebrated, but I asked myself, how much was actually deserved?
It was all deserved, Valentina, and more.
You know what I mean, Dylan. You know what I mean.
I know what you mean, but I'm inspired just to talk to you, because that word membership, that, to me, you are a real champion of membership and culture and just forward thinking and inclusivity, and the first person to say, I'm not doing enough, which seems crazy to me, but I think that really reflects your just dogged and, insatiable pursuit for a better whatever. I can imagine what it must be like to be a member in your lab. I want to finish the main part of the interview before we ask a couple of peripheral questions, circling back to you and your lab, specifically I would say unique and expansive approach to not just science, getting the curator of the British art in there on the paper because she has a unique view, but also nurturing scientists and citizens and leaders and members, right? Members of our common effort. One of the coolest things I saw on your website is the Inclusive Academia Journal Club, with topics including Asian Americans and the model minority myth, failure in science, love that one, privileges lead to inaction, among many others. Such a great forum to address issues that lie at the interface of society and the practice of science research, and also just like being a part of anything. I think any kind of group could benefit. I would love to be a guest. Might have to come up to Yale and check out that Inclusive Academia Journal Club. But what have you learned from this journal clubs? How do you think they complement your lab and the lives of those with it?
Yeah, so first of all, those are ideas, including the museum, for instance, was coming from a former student who told me, Valentina, they do these things. And then other people in the lab wanted to explore it, and Lauren Gonzalez in my lab championing that, and then organizing the workshop, writing about it. So I am so privileged and fortunate to work with so many different people. And so in the department 2020, all distorted by the yet senseless loss that we were now, especially white person like me, confronted to have to watch, whereas in a membership of colleagues and sisters that are from different memberships, they knew that reality, but we were feeling like we had to do something about it. So you see in the department now a catalyzing of discussion run by students and postdocs saying, faculty, student, we all gotta come together and read these papers and discuss it. They taught me a tool that then I could do in parallel to my lab and maintain over the years, because we live in a new heaven which we love, but in a Yale that is very much an ivory tower, when the reality of Valentina in Yale and Valentina outside Yale, meaning another person like me, just not within the Yale territory, is completely different. So here is a city that's given me an opportunity, a chance as an immigrant that doesn't treat everyone the same, and I get to go home and be safe at night in my bed because I have food on my table and others can't. So how do we reconcile that I hire within the ivory tower and that we are this customer of this city, which is amazing, but we don't get to know it, we don't get to have responsibility for it. For instance, right now we are reading this book, Yale and Slavery by David Bright, and we are planning to go to the museum and have that tour to understand the complicity we have in working in this lab, to the role that Yale had to say no to the first Black college in New Haven, right? So how can we do one thing but not the other, create such a dissonance within us? And so inspired by the department, by the younger generation who were telling us we gotta do something, the lab has become to practice their teaching every 6 weeks and organize in ways that initially was individually driven, but we learned soon that people felt taxed. People felt like when they were speaking about membership, that they were theirs but not shared, that they felt really vulnerable. And they decided, why don't we go in subgroups? So even the practice we have, it can be co-evolved and led by people in the lab in ways that, for me, is a new discovery of things that I would never have come up with, but been led by many others.
I think, it's just, as somebody who's a brand new PI or has been a PI for not too long, I look to folks like you as sources of inspiration in terms of how to run a lab that's, self-reflective, that's inclusive, that's able to incorporate the views from the most junior trainees. Because I think that hierarchy, like what you're alluding to, that hierarchy is unfortunately a reality in the ivory tower of academia. And I think a lab like yours really establishes a great model in terms of how to break down that potential hierarchy. Like I think we all aspire to that, where the greatest ideas can still come from a research assistant or an intern or a high school student. And I think your lab is a really great model system of how that could actually work. So like Dalen said, it's always so inspiring to talk to you, Valentina. It's a privilege to discuss science and policy and how to develop a more inclusive lab with you. I mean, I just remember coming out of the conversation that we had 4 years ago and feeling the exact same way. You're really an inspiration to us in terms of how to establish an inclusive, diverse, accessible stem cell community. And we're looking forward to your leadership for the ISSCR in just a few short months. Before we let you go, we want to ask you a few final questions, really quick peripheral questions. First of all, if you were not a scientist, and we're all very glad that you are a scientist and a leader in the stem cell community, if you were not a scientist, what would you be?
I will be, on the parenthesis, fail dancer.
Okay. All right. Very straight and to the point. I love that. What about the best piece of advice that you've ever been given, professional or not, building on all the amazing advice that you've given us today?
Lead with truth. It was from an organizational psychologist. I remember I was starting my lab and I was tiptoeing into how to give ownership to my student, but also make sure that I could share my own idea. I was afraid to be oppressive and so on. And I felt like this tiptoeing. Or in another setting, I remember I was really wrestling with that, admitting my vulnerability and weaknesses as a woman in particular, but in general, we have this portrayal of people in authority that know it all. They are self-sufficient. It couldn't be farther from truth, but that's the image and the expectation that we are all given. And so leading with truth and saying, my brain is chaotic. It starts with putting everything on the table and just being messy. And then I find order together with you, and I see what you pick and what I pick, and then we put it together. It was a really unconventional way to think about it. But as long as I let go of that vulnerability, weaknesses, and told the other person, you're strong in that, I'm not, can you help me and teach me? Those were literally undergraduate students or graduate students in my lab. Oh, a world of possibility open up.
Yes, I can only imagine how empowering that is. And yeah, I mean, you must create a really positive environment, because just here talking to you, I feel good about myself, Valentina. You're making me feel good.
That's the biggest compliment I've ever received, that you feel good about yourself. Isn't that a dream? Thank you, then.
Well, that's what I love about you. That's your goal, I think, in any interaction. You're so generous. And you've been very generous with us and our listeners. And I'm really looking forward to it. It's a good thing ISSCR is right around the corner, because now I get to see in person in just a few short weeks. And hopefully we can have another pleasant interaction. You can leave me on cloud 1,000,000. But
likewise.
Well, I hope I can return the favor. It's the least I can do for all you've done for me just in the last 30 minutes. Echoing everything Arun said, really looking forward to your presidency and the research coming out of your lab, and we hope to have you back on the show. We're gonna have to do like a reverse exponential here. It was 4 years, now we're gonna have to go to 2, and then we'll have you on every other week or so. But for now, it's goodbye. And thank you so much for sharing your vulnerability and excellence at the same time and joining us for this episode. All right, you guys, that brings us to the end of our show. Don't forget to subscribe to our newsletter at www.stemcellpodcast.com to get the show notes, including an episode summary and links to all the interview and Roundup papers. You can also reach out to us on X at STEMCELL Podcast or by email at info@stemcellpodcast.com with feedback or to suggest guests. By the time you listen to this, we'll be on the ground in Hamburg. Hope to see you. If you do see us tooling around the hallways, please come up, introduce yourself. We love talking to listeners to the show. We really stimulate our passion for science communication. Until the next episode, or those on-the-ground episodes that we're going to be coming up with over the course of the next week, thank you so much for listening.